Hello folks, Rodrigo Werle, weed scientist at UW-Madison. With me, I have Felipe Faleco, a weed science graduate student at UW-Madison. And today, we're here to talk about the concerning weed species, Palmer Amaranth, that's coming into our state via contaminated seed, grain, feed, and sometimes with equipment. Okay, so Palmer Amaranth used to be a major and driver weed problem in the Mid-South, and still is the #1 problematic weed species in the southern U.S. and is creeping into the Midwest, okay, becoming a major concerning problem. In the state of Wisconsin, thus far, we have detected this species in 9 different counties, okay. Last growing season, 2018, I was my first year here in Madison as a weed scientist, and I had the opportunity to visit 2 different fields that were infested with Palmer Amaranth. One of them, the introduction for the Palmer Amaranth seed was believed to be caused by feeding cotton seed from the south to dairy cows introduced to the field. And the second one was through contaminated grain that came to the state from other states. Okay, so at one of these fields at the end of the year, I had the opportunity to go back and collect seeds. We grew some of those seeds in the greenhouse. We then sent leaf samples to the University of Illinois Plant Clinic Lab for 2 reasons: to confirm that those plants were actually Palmer Amaranth and to test for resistance to glyphosate and PPO herbicides. Results came back and the population was indeed confirmed through their molecular test to be Palmer Amaranth and resistant to glyphosate. After we received those tests back, I challenged my graduate student Felipe to conduct the screening here that he's going to describe to us.
So here we are working with 2 populations. This population here is from western of Nebraska. It's a glyphosate susceptible population. And this one here, it's from southern of Wisconsin, that we are testing and compare if we found some resistance to this population. So we sprayed these populations with 8 different herbicides, glyphosates, and 7 more herbicides that is commonly used in corn production and soybean production. And we sprayed these populations between 2.5 and 4 inches, following all the label specifications and the adjuvants. And we sprayed these plants 20 days ago.
All right, folks, so Felipe just described the methodology for the study here, how we conducted it. And we thought it would be important to show and record this video here just because the results are quite impressive and not good for our production systems. Okay, so when I'm walking through the flats here, keep in mind that this population here is our control glyphosate sensitive from western Nebraska, and this here, it's always going to be the population from southern Wisconsin. As the molecular results from Illinois indicated, this population that we found in Wisconsin turned out to be resistant to glyphosate. We sprayed with the 22 fluid ounces of Roundup Power Max, and as you can see here, the plants are doing just fine, whereas we got complete control with our population from western Nebraska. Okay. The next herbicide that we conducted the molecular test for was PPO, or group 14. Okay, so we sprayed Cobra here post-emergence, and these are the plants that I have. So this population from Nebraska was also sensitive to PPO, or did not have the PPO mutation according to the molecular results. And here is our population from Wisconsin. We submitted the sample and it came back negative for that resistance. However, when we sprayed the 12.5 fluid ounces of Cobra, as you can see here, we could not get a complete kill. Okay, and that's the one important consideration when managing Palmer Amaranth and also water hemp. Post-control with PPO herbicides is very difficult. One, because of resistance. Some populations are resistant, but just because of the way this plant grows. And I want to expand that a little bit, showing this plant. So this plant here, my students chopped when it was a foot tall. Okay, and as you can see here, this plant is just growing back. So controlling pig weeds with contact herbicides post-emergence can be very difficult. Again, if the molecular results indicate you don't have resistance, that does not mean that the PPO post-emergence will work as effectively as you would like to. So here we have a tray, and these plants here were actually treated with Pursuit or Imazethapyr, a group 2 herbicide. ALS resistance in water hemp and Palmer Amaranth is quite widespread, and this is what we're seeing here. Pursuit sprayed post-emergence barely touched these plants when used at the label rate. Okay, Other two treatments I want to discuss. Here we have Atrazine, and here we have mesotrione or Callisto. Our Atrazine was sprayed at 4 pints per acre, and the Callisto was sprayed at 3 fluid ounces per acre. On the Atrazine treatment here, when sprayed post-emergence, as you can see here, the response is varying, but especially for our Wisconsin population here, we couldn't get a good or complete kill post-emergence. Okay, and a similar scenario here for mesotrione or Callisto. We could not get a complete kill. Even though these plants are highly injured, they're still alive. Okay, so remember, these herbicides were sprayed post-emergence to 2.5 to 4 inch tall plants. Felipe is going to conduct all the screenings looking at tolerance when these herbicides are sprayed pre-emergence, okay? Because this particular Callisto and Atrazine are commonly used in our state and around us as part of a pre-emergent herbicide program. So we want to see how these populations respond when used in a pre versus a post fashion. All right, so these are the 3 last herbicides I want to talk about, and these are very important herbicides for current production systems. Here I have dicamba, 2,4-D, and Liberty. Okay, these 3 herbicides can now be used in some of the novel traded technologies, your Extend, your Enlist, and Liberty systems. So again, it's, the systems are herbicide specific there. The first one I want to discuss is dicamba here. Okay, and as you can see here, even though we got good suppression, it was difficult to get a complete kill of the Palmer Amaranth. And a similar scenario for our 2,4-D, again, for both populations, the population from western Nebraska and also the population that we found here in Wisconsin. Okay, so both dicamba and 2,4-D are growth regulator herbicides, and even though they seem to be doing a decent job, we couldn't get a complete kill in there. Okay, the last one here is Liberty. And in these comparisons here, these 2 populations that I have, Liberty was the only herbicide that provided complete kill of our plants, so 100% control. There's a lot of interest in these traits. They can be helpful. However, our farmers have to be very wise how they use them. Okay, there's particularly a lot of interest around the Liberty technology. Is very effective on the pigweeds. However, if going that route, farmers must be spraying small weeds. And that's true for all herbicides post-emergence, but particularly for Liberty, which is a post-emergence herbicide. And through this point, scientists have not reported pigweed resistance to Liberty. Okay, so we got to use these herbicides, not only Liberty, but also the dicambas and 2,4-Ds, particularly in our soybean systems, in a wise fashion. Okay, so the take-home message here, we have Palmer Amaranth kind of coming into the state, and when it comes in with resistance. As we saw, glyphosate didn't touch it, imazethapyr didn't kill it, and it was difficult to control with the PPO. And then the control level with the other herbicides that we have here post-emergence was highly variable. Okay, so it's important to detect when the weed is present, adopt a sound management practice to prevent it from establishing in your fields and establishing around our communities here. So with that, thank you for your attention.